My name is Ann Meir, and I'm the founder of the Minnesota Lyme Association. Next month, we will have been together 19 years, so
Wow.
pretty proud of that. And welcome, everybody. We're from the Minnesota Lyme Association and the Greater Kansas City Lyme Association. We've joined forces. For over 30 years, IGeneX have been the global leader in the research and development of tests that accurately detect Lyme disease, tick-borne relapsing fever, and other tick-borne illnesses. IGeneX makes their singular mission to offer the best-in-class testing for tick-borne diseases that deliver the most comprehensive and accurate results possible, so patients can find the right treatment path to restore their health and get back to enjoying their lives. So with us tonight, Johan Humelius.
Very good. I like it. Hi, everyone. So I'm the local representative covering basically Upper Midwest for IGeneX. I'm based in Woodbury, Minnesota, so I'm very local to many of you. So, and then with me today I have Dr. Joe Burrascano.
Hello, everyone.
Hi, welcome.
Thank you very much. Man, you've been doing this so many years, so who's to say that Lyme really isn't a chronic disease, If it wasn't, you'd all not be here tonight, I guess. What would I suppose?
Dr. B, you're, at least to me, your sound is a little muffled.
All right, let me fix that. Sometimes computer acts bizarre. I have to then put on headphones. Bear with me a sec. All right, here we go. All right, how's this? Does this work?
I think it's better.
All right. I don't know, maybe some of this is just bad connection. Anyway, all right, so normally with these kind of presentations, I can get very technical and start to go into the weeds where I might lose a lot of you, especially this time of night. I mean, we're going to fall asleep. But I'm going to do something entirely different tonight. We're going to talk about how you get better, okay? And why does Lyme get to be so bad? Why is it so bad? What is it, and how do we fix it? So this is just an outline. I want to talk about some important features of these tick-borne disease infections, the immune system, the effect on the blood vessels. It gets inflamed. They call it a vasculitis. Biofilms, which are like gelatin-like substances that the tick-borne disease germs make, the toxins that they're involved in chronic Lyme, the problems with energy production, all those are the basics that we have to go over. Why are there neurologic manifestations? That's important because there are a lot of different theories, and a lot of them relate to Lyme and the co-infections. I want to go through some of the details of the different germs that are involved, give you a better idea about it. And then when it comes to testing, the modern take on testing, why things have changed, why the standard big laboratory tests are no good anymore in light of the new diagnostic testing from IGeneX and others. Then we'll spend a bit of time talking about how to get better. And this is probably something you've not heard before because it's all the details that you might have heard about, like toxins and molds and all that. So we'll go through it. We'll start with the immune system. Turns out that Lyme germ itself inhibits the immune system. In our immune system, there are B cells that make antibodies, and there are T cells, and they're like the pac-man that gobble up the germs. And it's found that Lyme germs, if you put them in a test tube with your blood, they can impair and decrease the function of both B and T cells. And actually, some even get killed by the Lyme. Also, the killer cells, that's a CD57 test. When Lyme is present, the killer cells can't get mature. You get a very low count. The immature cells are there that don't work as well. Turns out when Babesia with Lyme, it's much worse impact on the immune system. And now we're finding that Bartonella probably does the same thing. So if your immune system is impaired, what does that mean? Well, you get a negative blood test because these blood tests are based on antibodies made by the B cells. So if the B cells are not functioning or there are fewer of them, you're likely to get a negative blood test. If your immune system's impaired, you have a hard time getting over an infection. And that can be one of the reasons why Lyme and all the others are so chronic. It also means that, we all harbor other germs that are usually kept at bay by our immune system. If our immune system is weakened, these latent infections can reactivate. Finally, we also have other germs in our system that are kept at bay that we don't want them to overgrow, but when the immune system's down, you can get these things that are there in small amounts to overgrow, and the main example of this are yeast germs in the GI tract. So this is vicious cycle of impaired immune system, present viruses and yeast, and then more viruses. So what I mean is that if you're sick with Lyme to the point of being chronically sick, which definitely after 1 year, maybe even less than that, the viruses that are latent can reactivate. Okay, so the most common one is the Epstein-Barr virus, the one that causes mononucleosis. In the adult who's already had mono, it can reactivate. You get fatigue, sore throat. In the throat, you can find little red streaks on the tonsils. We call them crimson crescents, headaches, aches. And maybe there's really profound fatigue. You can wake up from a night's sleep and wake up tired, wish you can have a nap. Other viruses like herpes 1 and 2 can become reactivated. Another germ, a virus called Bourne virus, is well known in Europe and not much talked about here. But that's a virus that can become reactivated and cause treatment-resistant depression. That's not talked about much, but it's actually kind of easy to fix. And other virus called HHV6 is like Epstein-Barr, can make you have that chronic fatigue type thing. In our intestinal tract, there are yeasts that live there all the time, but they can overgrow. What they do is they adhere to the lining of our gut and they produce a dense biofilm. It's like a gelatin substance. And inside that, not only are there yeast germs, but there are viruses. And a recent study showed that the viruses like this jelly, this biofilm, it protects them and allows them to really grow and actually can make the viruses worse. So the funny thing here is that you have a yeast problem that makes a virus problem. All right, now, any of these viruses can contribute to the fatigue, the nerve problems, and the brain fog. The other thing is that hidden co-infections may become apparent. A lot of times people, let's say they have Lyme, and they're sort of getting better, they're not as bad as they were, but they're not really getting 100% better. And then the immune system starts to run down, and turns out they had Babesia too, and they didn't realize until the Lyme weakened them and they start to get Babesia symptoms. Mycoplasma is another one that can get reactivated, and that's also immune suppressive. So there's also a flip side to this. People with long COVID or unexplained chronic fatigue, especially if they have activated viruses, you should take a step back and say, okay, what is the cause of this? Maybe there's an underlying trigger like a tick-borne disease or toxins or other things. So let me talk about some other things. With Lyme and Bartonella, one of the big things they do is they cause inflammation of the blood vessels. That's called
stinky.
What's that?
You're like,
Somebody, your microphone.
if we can have everyone mute themselves. And then also, Kurt, do we have anyone waiting in the waiting room to come into the meeting? I just heard that we have a couple people waiting.
I didn't even know we had a waiting room on Zoom.
I didn't know that either, but
Do they have to be admitted?
She's on.
oh,
She-
okay.
She got on. Thank
Okay,
you.
thanks.
Okay, so let's go on. So Lyme and Bartonella cause inflammation of the blood vessels. That's called the vasculitis. And when that happens, the blood vessels swell and get inflamed and can choke down and impair your circulation. They can cause more inflammation elsewhere and poisons and so forth. Lyme and Bartonella also make biofilm, and they are not only in the tissues, but actually floating around in the bloodstream and in some cases actually plugging the circulation. Babesia does that too, but in a different way. It sludges and makes a biofilm, but also causes clots, they call them microclots, in the veins and maybe even the arteries too. And the rickettsias like Ehrlichia and Anaplasma Rocky Mountain and so forth, they also cause vasculitis and they affect the blood count as well. It's been found that ticks carry worms called nematodes. It's not clear whether they're transmitted with a tick bite, but if they are, the problem is they also make biofilms. Okay, so you're seeing a pattern. It's vasculitis and biofilms, poor circulation, sludging, clots. So it's really hurting your circulation, especially when you have co-infections with the big three, the Babesia, Bartonella, and Borrelia. When you have the bad circulation, that's the main reason why you get brain fog. People talk about, a PET scan or a SPECT scan will show decreased blood perfusion and so forth. Well, this is exactly what's going on. In Babesia, people often get a strange sense of being imbalanced, not really vertigo, but kind of lightheaded and feeling like they're going to fall. That's because of poor circulation in the brain. In Babesia, often there's air hunger where you feel like you can't catch your breath as well. Same thing. And POTS, the postural orthostatic thing where you stand up and your blood pressure drops, again, circulation. So poor circulation is why there's poor stamina, weak muscles, soreness if you even exert yourself a little bit. They get less nutrition, the muscles and nerves, and the waste products like lactic acid build up. And even digestion gets affected because we need a lot of good circulation to help digestion go along, and that doesn't always happen. And what about toxins? Why do they build up in people who have Lyme and other tick-borne diseases? Well, chronic inflammation from any source impairs toxin clearance, okay? There's a compound that our body makes called glutathione, and that's the main chemical in our body that removes toxins. So when you have a lot of toxins, this glutathione gets used up. Other thing is that, these infections impair our metabolism. They slow down the conversion of food into energy. And one of the results of that is that we don't make as much glutathione. So we're using it up faster and we're not making as much. So you get depletion of this glutathione. And because that's what gets rid of toxins, we don't have enough glutathione, the toxins build up. There's a thing called methylation in our metabolism, the steps that bring our food with oxygen into energy. There's a part of that cascade that's called methylation. And some people have a genetic defect in this. It's called MTHFR. And if they have this genetic defect, methylation is slowed down, and methylation is what makes glutathione. Okay? It turns out, even if you don't have this abnormal gene, even people with normal genetics, because of the Lyme, you get inhibition of this methylation process. So again, impaired energy because you're not metabolizing. Your immune system is down because it uses energy for healing. And the glutathione, again, is not being produced. Another thing that happens, this is a long list. Bile flow from the liver is decreased. Bile carries the toxins out of the liver because the liver is like the main filter of the body, and then the toxins get put out through the bile into our intestinal tract and down the drain they go. But the problem is the bile flow is impaired and the toxins don't come out. And finally, all the inflammation and infection causes leaky gut, and some of these toxins, which should go down the drain when we go to the bathroom, don't. They get reabsorbed. Another big thing I want to spend some time on is the mitochondria. Inflammation causes mitochondrial dysfunction and leaky gut. And I say, what's the mitochondria? I'll tell you about that in a second. All right, what about the toxins? Which ones build up? All of them, okay? Most common, of course, are the mold toxins, because they're all throughout the environment. Not just maybe in a water-damaged house, but they can be in the fields. I live in a damp area, and I go out some days, and I can just smell the mold in the air. So normally our body gets rid of it, but with these infections, we can't. So the mycotoxins build up. Toxic metals. I found out that chocolate has cadmium in it, of all things. Some coffees have other things. So toxic metals can build up. The weed killers, the Roundup, the insecticides, plastics, volatile compounds, all these things can build up. And the interesting thing, and the thing you have to understand, is that most of these toxins, when they're in our system, they're there permanently. No matter how much antibiotic you take, no matter how clean you're eating and how detoxified your house is, if you have toxins, most of these will not go away without treatment. Now, what do we do? Well, they're a little bit fat soluble, at least to some degree. And that's important because in our cells, we have mitochondria. They're the things that produce the energy. That's where the food turns into energy, these little organelles. And they're made of little blobs of fat. So these compounds, which are dissolved well into fat, they're going to concentrate there and impair the mitochondria and even kill some of them. The metals and other toxins can be directly toxic to the cells themselves. For example, nerve cells, they get from metals, for example, brain dysfunction, neuropathy, the bone marrow affects our blood counts, our immune system. In the GI tract, our digestion is affected, more leaky gut. And the toxins also can kill the good germs in our intestinal tract and cause more trouble. So here are the mitochondria. They are in our cells as little organelles. The top picture here you see is a diagram of what they look like, and the bottom is actually a photograph inside of a cell. These little things, they contain folded membranes. That's what these things are, the little folded membranes. These membranes, again, are made of fat, okay? And it turns out that this metabolism, turning the food with oxygen into energy, it doesn't occur like in the soup of the liquid in the cell. It doesn't. It occurs on the surface of these little membranes. So if these membranes, which are made of fat and absorbing toxins, are affected, that's why the metabolism slows down. That's the fatigue. That's immune dysfunction. That's all the things that happen with these tick-borne diseases. So any damage to this, from toxins, from other things, is going to cause problems. The mitochondria also have their own problem they call cell danger response. If they feel a lot of inflammation going on in the body, they shut themselves down on purpose to protect themselves. It's kind of a form of hibernation. And that's another reason why people with chronic infections, especially tick-borne ones, have mitochondrial dysfunction. So remember the mitochondria, that's really, really important. Now, if you're not making energy, they have big effects everywhere. Obviously, things like fatigue plus stamina weakness if you're not making energy. But other thing is, there's massively impaired immune function. The immune system takes a lot of energy to run. The brain is also the most energy-hungry organ in the body, and the brain fog is another manifestation of that, and obviously poor healing. So there's a cascade. I made this, so I'm proud of myself because I made this. So you have the infection. The infection causes inflammation. The inflammation causes the toxins to build up because it doesn't make glutathione. The toxin buildup decreases your energy production because it affects your metabolism. All of that poor metabolism impairs our immunity, which allows infections to flourish and become chronic. Now, all these other things by themselves, for example, inflammation can impair immunity, toxin can impair immunity directly, decrease energy production obviously does. So all of these things circulate and make the Lyme person sicker and harder to treat and less likely to get better. Now, what about the neurologic things? In Lou Gehrig's disease, ALS, the theory is that the germs or pathogens cause this impaired metabolism in the nerve cells that run our muscles and eventually the entire nervous system. And the thinking is that if you can kill the pathogens or at least suppress them and rebuild the metabolism, you can stop the disease from progressing. There have been a number of examples of this. Not every case, but in a lot of cases, the majority. Alzheimer's, it's found that it's a direct infection of the brain by different bacteria, especially by spirochetes. That's been shown by 2 different researchers. Other things have been in there too, like toxoplasma can be doing that in others. And with multiple sclerosis, the theory is that the infections and inflammation directly damage the nerves and the myelin, which is the insulation around the nerves. And of course, the infections cause inflammation of the blood vessels, and those don't nourish the nerves. Biofilms, sludging, microclots, other things I talked about before. And interestingly, studies done in Europe and now done here show that in the great majority of patients with MS, as many as 90% of them have blocked veins in their neck. And the poisons and the toxins that build up in the brain are not able to drain. And they had studies done where they actually used balloons to open up these veins, and the people's MS symptoms immediately stopped, but the veins immediately blocked again. So there's some other process going on. And again, probably biofilms and vasculitis that cause this to keep happening. Now, not everybody with Lyme gets MS or Alzheimer's and ALS, so there obviously are other components, there's genetics and environmental factors and so forth. But you need all of these together, just the genetics and not the infection, you're not going to have trouble. The infection and not the genetics, you're not going to have as much trouble, but you have to have all of it. All right, so what do you have to do here? Obviously, you have to accurately diagnose the presence of these tick-borne diseases. You have to test all the different toxins if you can. Then design a very carefully crafted treatment plan. And as patients, you all need to keep a daily diary because the whole thing about treating Lyme and tick-borne disease is called pattern recognition. So keeping a diary helps the clinicians and yourself spot patterns, identify these patterns, and look for trends. So that way, can help to craft your regimen more carefully. Another thing which I'm very big on is always schedule a follow-up visit. I can't say how bizarre it is. You go to a doctor, go all through the thing, and say, okay, bye. Well, what about follow-up? The Lyme patient you really have to insist on regular meetings with the practitioner about always scheduling a follow-up appointment every time. Don't wait and say, oh, I'll give them a call when I need them, because by then they're already busy, you're not going to get an appointment for a few months. So always remember to get a follow-up visit. All right, know your pathogens. In Lyme, there's not just one Lyme germ, there are many Lyme germs, probably more than a dozen. Tick-borne relapsing fever, you know, there are many of these germs too, and people don't often talk about that, but they should, because tick-borne relapsing fever is caused by a germ like Lyme. It's also a Borrelia, and it causes symptoms just like Lyme. In fact, you can't tell them apart in some cases, but these are different germs. They're not picked up by Lyme tests. How many Lyme patients are sick with what looks like Lyme? The blood test shows nothing, and the doctor throws them out of the office. Well, they're testing for the wrong germ, because relapsing fever could be there, and the regular Lyme tests don't pick it up. Bartonella is a huge problem. Every time I look it up, there are more species found. We have at least 45 that we know about, possibly over a dozen infect humans. Babesia, we know there are at least 5 species in America, and this may be just the tip of the iceberg because more are being found. The Rickettsia family, Anaplasma, Ehrlichia, Rocky Mountain, they're the most common group of germs if you look at ticks. If you go into the woods and you collect ticks and analyze them, the number one, no matter it's America, Europe, Asia, are the Rickettsias, the Anaplasma, Ehrlichia, and other related germs. And the truth is they're probably being underdiagnosed. Everybody thinks about Lyme. They don't think about these, but these are there too. Tick-borne viruses, there are many of them, and some can cause you to get really, really sick, but they're usually not chronic unless your immune system is really low. And like I mentioned before, nematodes are worms, actual parasitic worms that are found in ticks, and we don't know whether they get into people. So that's a big question mark. Now, remember I said know your germs. This is a list of the different Lyme germs. On the left is the Borrelia, the Lyme Borrelia. There's Borrelia burgdorferi. This is what people know about as causing Lyme. But Borrelia burgdorferi, which is known as Borrelia senso stricto, strain B31, that's just one of all these different germs that can cause Lyme disease. The problem is, the reason why I made this in red, if you go to a place like LabCorp or Quest to get a Lyme test, they're only going to test you for this one. They're not going to test you for any of those others, even though they're always present in America and in Lyme patients. These are the relapsing fever germs. Again, these two in red, you can go to a lab like Quest or LabCorp and order a test for one or the other or both. But all these others have been found in American patients, causing Lyme-like symptoms and relapsing fever type symptoms, and they're not going to be picked up if you don't get the right testing. In fact, you can't even be tested for any of these by any other lab but IGeneX. Okay, IGeneX will test for all of these. When you order a Lyme test, you're getting all of these. When you order a relapsing fever test, you're getting all of these. Okay, so that's a major, major difference about IGeneX versus other labs. If you're going to be tested, use IGeneX. I'm not just saying that because they're sponsoring this meeting tonight, but as a clinician, what if you have one of these, non-specific or non-common germs, and you get tested for this? You're not going to get picked up. You're going to say, well, you're having stress, or maybe you're just tired, and you've heard the stories. So again, insist on the better test. Insist on the test that will look for all the different species. Now, what about Babesia? Same story. You can get a blood test for Babesia microti. If you go to the big labs, say, I want a Babesia test, they're going to test just for that. You can ask for Babesia duncani, which is probably even more common, but not all labs offer that. But all these others on the list are known to cause babesiosis in humans. When you get a Babesia test from IGeneX, all of these are included. If you get this from a big lab, you're only going to get one or the other and not any of the others. Okay, so again, critical if you're going to do a test, get the right test. And here comes Bartonella. I mean, this is, I think 17 or 18 on the list. You go to a big lab, you can get Bartonella Hensley and Quintana. All these others are infecting people, causing sometimes catastrophic illness, and you won't get picked up. Again, IGeneX, when you test for Bartonella, all of these come out. Okay, if you have them, they're going to find it. Another thing I want to tell you about, there's a big myth out there that the tick has to be attached to you like overnight or 36 hours. It's absolutely not true. Okay, those studies were done on rabbits, which don't really get Lyme disease, and on rodents who have to have immune system genetic alterations so you can get Lyme disease like humans do. So it's based on fantasy. William Bergdorf himself, he said that 5 to 10% of ticks with Lyme have it in their saliva. Now, what does that mean? When a tick first bites you, the very first thing they do is they inject their saliva into your skin, because the saliva is an anesthetic and it deadens your skin so that you don't know you're getting bitten. And you get bitten by like a deer fly or a horse fly, you feel the pinch, you smack it. But with Lyme, you don't feel the tick bite because of the saliva. So again, the first thing they do is they inject saliva. Now, if you have germs in the tick saliva, those germs are going to get injected before the thing even gets engorged, okay? So Will himself said 5 to 10% of ticks that he's looked at have Lyme in the saliva. Relapsing fever, not only is it in the saliva, there's been a test where you can find that, or a study, transmission in as little as 1 minute. In fact, one study, it was 15 seconds, okay? Bartonella is also found in tick saliva. So again, rapid transmission is probable. The rickettsias and even arbovirus, these are the tick-borne viruses, are also present in tick saliva. And babesia, the babesia get in the salivary gland of the tick before it even bites you. So again, all these major infections can occur within minutes of the tick bite. So don't say, well, the tick wasn't on me overnight or 2 days, I don't have to worry about it. That's absolutely not true. Other two things, if the tick is not properly removed, it can actually cause infection, because if the tick is engorged and you pinch with your fingers and pull it out, you're actually squeezing it like a tube of toothpaste, and you're squeezing the germs into the body. So that's not good. If you put heat on it or chemicals, it causes a contraction of the stomach and it squirts even more germs into you. So you can't do that. You have to get a very fine tweezer and pull it out from the mouth. And don't just pull it straight out like a splinter. You put pressure on, you pull some traction on it, and just keep pulling for 30 seconds, a minute, maybe even 2 minutes, and little by little it'll back out. If you pull it out too quickly, it'll break off, and then you have some infected tick parts in your skin. That's not good. Another thing is not nice to think about. Ticks drink a lot of blood, and what goes in one end comes out the other. And it turns out that tick feces are very highly infectious. And if you have a dog or a cat where there's been ticks on them, and you look at where the tick is in the skin, all around the hair, you'll find this little black material, and that's the feces of the tick, and it's very highly infectious. And that's why, number one, you don't touch the tick with bare hands. If you have to touch it, you put a glove on. It's why you handle it with a tweezer. It's why after the tick is out, then you put antiseptics on the wound and the skin around it. And if it's one of your poor cats or dogs or other pets, and it's got this feces on, you really have to give them a good shampoo. Okay, we're going to talk about testing. Just like you have to know the germs, you have to know the testing itself. There are two kinds. There's an indirect test and a direct test. Okay, what does that mean? An indirect test doesn't actually look for the germ. It looks for evidence that the germ is in your system. What do I mean by that? It means in our body, if we get infected, our immune system recognizes this infection and makes a response. Okay, so the indirect tests measure that immune response. The problem is it shows that you've been exposed to the germ, and that could have been exposure yesterday or a month ago or 2 years ago or 10 years ago. So you don't really know if that means your infection is present right now. It just shows you've been exposed. Next thing is that because it's a measure of immune reactivity, if your immune system's depleted, as I said happens, then the test becomes less accurate. When you do a blood test like a serology, which is a Western blot, an immunoblot, all those others, the measuring response of the B cells in your bloodstream. And that's the quickest and easiest one to do. But of all these tests, only the immunoblot from IGeneX is recommended, and I'll show you why in a second. The other test of the immune system response is not the B cells, which are serologies, but the T cells. And IGeneX offers a test called the IGeX spot. So there are actually two different ways of looking for the body's reaction to a germ. And clinicians often do both, not only to help diagnose it, but then they get a good picture of whether the immune system is working the way it should. Okay, the other kind of test is a direct test. It looks actually for the germ itself or maybe parts of it. Now, if it's positive, the beauty of that means that the germ was present the day the blood test was taken if it's a positive. The problem, though, is that a lot of these germs either don't live in the bloodstream or they're stuck in biofilms or some other place, and they're not always in the blood. So these direct tests are very, they're not that sensitive. In other words, it's like going fishing. If you don't catch a fish, it doesn't mean you don't have fish in the water, but it means you just didn't catch one. If you catch a fish, then you know there's fish there. So the same analogy applies here. When you're doing a direct test, It doesn't always show up, but if it does show up, you know you definitely have it. Now, what's a direct test? Well, the FISH test, a fluorescent in situ hybridization, a FISH test, IGeneX offers that. It's a direct test where the blood's put on the smear or glass slide and stained with a dye, and it looks for the germ. Antigen capture is a way of looking for components of the germ in the urine. And culture is where the blood is taken and incubated in the lab for a couple of weeks, and then it's tested usually by a PCR. Another direct test, the PCR itself, a lot of you have heard of that. The problem is PCRs don't work well in the blood. They pick up, I know in Bartonella literature, it only picked up 6% of infections. So in other words, you get a 94% false negative rate. Now you get a positive PCR, you know you're definitely infected, but a negative doesn't mean you don't have it. So these direct tests are very good for being specific, and if you have a positive, you believe it. But if you have a negative, it doesn't mean you're not sick. They just don't pick up all the infections. A little more details on the T cell test. T cells are part of the immune system. Like I said, they're the Pac-Man that eat up the germs. They're also responsible for memory, immune system memory. So if someone gets infected, the T cells, being part of the immune system, they activate and recognize the infection. So the test involves taking a blood sample. In the laboratory, they separate out the T cells and they put some of the germs in this solution. And if the infection had been present, the T cells, which are now primed from the infected patient, they'll activate, and the test measures that activation. What IGeneX does one step further, they don't look for only like Borrelia burgdorferi and Borrelia miyamotoi. They look for all Borrelia. They look at a genus level, not at individual species. So it's much more sensitive than the average test because it looks for all the different germs. Like I showed you in that a few slides ago, it's not one germ, it's dozens of germs in each category. The other thing about the T cell test, that it doesn't always work. And I mean that from a time point of view. It's very useful early in the infection because T cells wake up right away. But after a few weeks, their sensitivity decreases. And they're usually not good in disseminated Lyme until you get into the chronic phase. And then when you're really, really chronically sick with Lyme, then again, the sensitivity improves. So it's only useful in very early disease and very late disease. And in between them, it's probably not worth doing. Now, in terms of the other indirect tests, these are the serologies, the IFA, the Western blot, and the ELISA. They don't recommend them anymore. I don't recommend them anymore because they only look for that one species I showed you in the slide in red. And the way it works is, if you're exposed to an infection, the B cells activate and they produce antibodies, and the test measures antibodies. What they do is they measure the antibodies in the circulation. Sometimes though antibodies don't circulate, they get bound up in the immune system and they're not free to run around, and that's why sometimes you get a negative test. And the reason why IFAs, ELISAs, Western blots are not recommended, number one, they're very insensitive. If you go to, let's say, Quest or LabCorp and you want to get a two-tier serology, which is an ELISA and a Western blot, or an IFA and a Western blot, or two ELISAs, number of different schemes, the sensitivity of that is about 50%. If you get a good Western blot, it might be as high as 70%. So, you're missing a third to a half of the cases. So that's really silly. And the IFAs themselves, which still are being done by a lot of labs, a lot of false positives. Other germs in your system can make it look like you're infected with Lyme, for example, when in fact you aren't. So an IFA test is just old-fashioned technology. Another big thing, they only test for that one species. Again, remember that list I showed you, the ones in red are the ones that these tests for, and all the others are ignored. So that's a good reason why not to do the test. Now, Vibrant Lab uses a microarray, they call it, and it's a modification of the ELISA. And in the unpublished studies, it was about as sensitive as a two-tier test, like 50 or 55%. So that's also not really recommended if you want to know what's going on. The CDC has come up with ways of diagnosing Lyme, and they use the Western blot. Now, what is a Western blot? Well, it's an antibody test. And what they do, they take the germ, the Lyme germ in this case, and they break it up into millions of little pieces. They use sound waves to sonicate it and break it up. And then they filter them and spread them out on paper using an electrical charge. It's called electrophoresis. So if you have antibodies, if your immune system has recognized the infection, the antibodies will form what's called a band. You see the dark dots in the picture, these dark spots, these are called bands. And the bands are given numbers based on how big they are, believe it or not, their molecular weight. And what the CDC did is that they said, okay, when you interpret a Western blot, we're going to say that there are 10 bands you have to look for. And these are the ones, band number 18 and 23 and all so forth. And to be positive, you need to have 5 out of these 10 to be positive. If you get 4 out of 10, you don't have Lyme disease. Now, that's ridiculous. Not everybody has 5 bands. If someone is sick with Lyme and you have all the symptoms of Lyme and you have 4 bands, does that mean you don't have Lyme disease? It's silly. Also, if you look at what these bands represent, 7 out of the 10 are not even specific to Lyme. Like, for example, band 30 is found in some viruses, as band 58 is found in some germs of your intestinal tract. 41 is found in the mouth, germs of the mouth. So, as silly as it is to have this arbitrary 5 out of 10, 7 out of the 10 are not even specific to Lyme disease. Turns out the ones that are specific to Lyme, bands number 31 and 34, are not included in this. It was a political thing having to do with the Lyme vaccine back in the day. So whatever it is, that has persisted. And the last thing, again, just like the other serologies, it's based on just that one strain, the B31, that one I showed you in red, and none of the others. So again, that's why this isn't so accurate. You know, these bands that you show up here, they're like ink blots. They're not always exactly readable. This here shows the what's called the control, the test strip, and this tells you, right, this dot is this band. So you're supposed to line up, and they don't always line up. And a dark band is obvious, but what about a pale band? Do you count that? You know, just a lot of guesswork, not very accurate. So what IGeneX did was they completely threw all that out. They just got rid of it all, and they made a thing called an immunoblot. I know it sounds like Western blot, but it's absolutely completely different. Instead of being based on a lab strain, like a Borrelia germ, like I showed you again in that red list on the list, they take the antigens individually. Now, again, with the old-fashioned serologies, they take one germ, they break it into a lot of pieces, and they spread that out on that paper to make those bands. But the problem is it's one species. When you release antigens from a broken up germ, a lot of non-specific ones come out like I showed you. So you're not getting much accuracy there, and it's only one species. So what IGeneX did was they went the other way. They made these reagents, these chemicals for their immunoblot synthetically in the laboratory. They actually cloned the genetics of the Lyme germ and all the other germs they make immunoblots for. But they, in this case, they cloned the antigens from the genes of the Lyme germ, and in the lab they use the genetic code to create the exact antigen they wanted to make. And instead of spreading it out on paper with electricity and all, they actually print it using a printer, just like an inkjet printer at home. The result is it's much more sensitive, much more specific, and it'll pick up all those different species, not just that one in red. And as a result, it's the highest sensitivity and specificity of any Lyme serologic test. The sensitivity in Lyme ranges based on how you test it, anywhere from 90 to 100%, not 50% like the Western blot. And the specificity is between 97 and 100% based on which test you're doing, including the IgM. Okay, what does that mean? Well, first of all, as I said, it's multiple species, not just the one. It's so sensitive that it's the only test that you can use in early Lyme disease. The sensitivity is between 83 and 93% in early Lyme. And again, the dogma is you can't test someone for Lyme disease when they first get sick because it's not long enough to make these tests show. In this case, you can use an immunoblot. Now, in some people's Lyme, they don't get the standard positive test they get, which is, I will explain this simply. When you get an infection, we make an antibody called IgM that starts right away in the first week or two and then disappears after a month or 6 weeks. Then a different antibody forms called IgG, and that persists during the course of the infection. Now, in some people, in some Lyme patients, the IgM doesn't go away after a month. It stays there. And in fact, in some cases, the G thing, which is supposed to be after the IgM goes away, IgG never appears. So a lot of the blood tests, again, CDC sanctioned, are based on IgG. And if they say, well, IgM should go away, if you still have IgM, don't believe it, it's a false positive. Well, in the case of an immunoblot, it's not a false positive because in late disease, even the IgM alone has a specificity of 98%. So this is the only test out there, again, where you can believe a positive IgM. I know I'm getting a little too detailed here, but I just have to give you examples of why this test is so much better. And it's not just Lyme, it's also relapsing fever, Bartonella, and Babesia. Now, just to give a comparison, the Lyme Western blot, they take a germ in a test tube, they grow it, they break it into a lot of little pieces, and they spread it out on this paper with electricity and try and match it to the control. The immunoblot, they make these lab-created antigens. They use a printer to put them exactly where they want them, so they know what they're looking at. It's not just smeared out like this. And the darkness or lightness is based on the person's response, not on whether or not it's a good production run in the laboratory. So it's a much, much more accurate test than the standard one. Again, I'm going to repeat this. This is, again, why testing for multiple species is so critical. The ones in red, that's what you can get from Quest and LabCorp. All the others are infecting people, and all the others can be picked up by IGeneX. Okay, lesson learned. Now, another nice thing is that this immunoblot for Lyme is now cleared by FDA. They are marketing it as the iDart immunoblot. Now, the nice thing about this is not is it a very accurate test, as I explained, it's also a two-tier test. So that includes the first test, which is called an LSA in this case, is tier 1, and immunoblot is tier 2. More importantly, it's also based on the IGeneX criteria. The CDC criteria is what they used to use on the Western blot. And like I said, they have 10 random bands, you have to have 5 out of the 10. Here, IGeneX uses just 2 band groups, not 5 out of 10. None of the non-specific bands, so 7 out of 10 that I showed you, the non-specific, none of them are on the immunoblot from IGeneX. But the bands 31 and 34, which were excluded from the Western blot, are now part of this. And this is the only FDA-cleared Lyme test that includes these bands. So one more reason why this is such a better test. In fact, perfect example, this test is so accurate, it forced the CDC to change its webpage. The webpage used to say a positive test is a Western blot with 5 out of 10 bands, blah, blah, blah. Now it doesn't say that at all. Okay, it's no longer saying you have to have 5 out of 10. They're not listing all these non-specific bands. They're not excluding band 31, 34. Basically, they're saying if you have a positive from an immunoblot, you've got a positive for CDC. So this is a gigantic win. We've been fighting CDC since the mid-1990s to try and get them to change. And finally, they had to change because technology showed them what to do. Now, a little bit of semantics here. Immunoblot, which is what IGeneX calls their test, that's basically an umbrella term. It just means a laboratory test that analyzes specific proteins, okay? So technically, a Western blot is a form of an immunoblot. In fact, I looked up at LabCorp, they call their Lyme test an immunoblot, when in fact it's a Western blot. Because an immunoblot, like I said, is a generic term for this kind of a test. A Western blot is one form of an immunoblot. But IGeneX is the only one that offers an immunoblot that's based on these laboratory-created germs that's also created to look for multiple species. And that's why, again, immunoblots are not created alike, and only IGeneX makes this kind of an immunoblot. Also, speaking of testing, I want to tell you about the AccuDart. I don't know if you've heard of this, but it's a home blood test kit that was developed by IGeneX. It uses the same technology as the immunoblot, these recombinant lab-created proteins. And it's a home test kit. You buy it online, accu.health.com. You get a kit, you do a fill the stick, you put it in a box, and mail it back to IGeneX for processing. You do not need a prescription for this. Any of you can just order this. You don't have to get a doctor to order it. And that's great. The sensitivity of this is 83%, much better than the CDC's testing. And it's the only test better than this is immunoblot itself. And you can get this for Lyme, relapsing fever, Babesia, and Bartonella. So, I mean, think about it. You don't have to travel to a lab. You can get this done before your first visit, between visits. If there's telehealth, you don't have to say, where am I going to go? Just order it, get it done at home. If you have a family member who says, I don't have Lyme, leave me alone, and you say, I know you got it. So you order a test, you get the test done, 1, 2, 3. And you can always, if you have a positive test, the one you're not sure about, you can always just order an immunoblot to get confirmation. So this is a real big deal. You should know about it because it's a real test. It's a good, strong, positive type of test. I want to talk about the direct test just briefly. Like I said, they're not very sensitive, but they are very specific. The PCR looks for fragments of DNA, the genetic material of the germ. In blood and spinal fluid, like I said, it picks up 6%. In the urine, it's a little bit better. In biopsies, it picks up maybe half. So a positive is great, but don't always expect one. The FISH is a fluorescent in situ hybridization assay, a FISH. They take a drop of blood, they put it on the slide, and they look at it under the microscope. The trick here is that they don't just use a dye like most labs use an ink-based dye, believe it or not, when they look at your blood under a microscope. Here, it's a very high-tech thing. They use a kind of a dye. It's not really a dye, but they use a dye that actually binds to the genetic material of the germ, or the RNA in this case. And then they have it attached to a fluorescent marker. So when you look at this under the microscope, number one, it only goes to the germs that have this genetic material. And number two, it fluoresces. So you can see it much more easily than something stained with ink. And in fact, the published studies show that for Babesia and Bartonella, it's 100 to 1,000 times more sensitive than a basic blood smear. And the importance of this is that if you go to the hospital for a Babesia test, they're going to do a standard blood smear, and that can pick up maybe 1% of the infections, where this picks up 100 to 1,000 times more than that. And finally, the other direct test is the culture. That's where the blood is taken in the lab and put in an incubator for a couple of weeks, and then it's tested. And IGeneX is the only lab that offers that. That's for Lyme, relapsing fever, also Babesia, Bartonella, Ehrlichia, Anaplasma, and some of the other rickettsias. Now, why is that so good? First of all, when you culture a germ, remember I said like the PCR, the other direct tests are not very sensitive because there are very few germs. Well, by putting in the culture, letting it grow and multiply, now you can pick them up. When they test it, again, it's at a genus level. So they look for Borrelia, or they look for Bartonella. They don't look for individual ones. They look for the whole group, and that way you don't miss any of them. And they found that when you do this test, it's 100% specific. In other words, if they say Lyme is there, you know you got Lyme. If they say Babesia is there, you know you got Babesia. And the sensitivity is at least 10 times that of a plain PCR. So it's a much, much better way of doing a direct test. Now, I showed you the slide last time I spoke to you. With this culture, they found really bizarre germs. One germ called Anaplasma platus is an infection in dogs, never expected to be in people. So they found this in a Lyme patient. And in the worldwide literature, when you look up the cases, there's only 4 cases reported in the whole world of this, and IGeneX happens to find case number 5. Another one, which is even more wild, is a thing called Colpodola. There's a chronic fatigue patient, a lady in her 40s, who was sick for years, and no one could find out what was wrong with her. None of the Lyme tests or anything showed anything. So she had the culture done, and they found a thing called Colpodola, which is it's like a protozoa that lives in ponds and in soil, and sometimes they're identified in animals. And this woman had this in her blood. Now, they didn't believe it. So, the lab, IGeneX didn't believe it. So they sent it out to get sequenced. And sure enough, it was Colpodola. So they didn't believe that. And they did the second time, and it was Colpodola. Only 2 cases reported this infection in the whole worldwide literature, and they found one. So they had case number 3. So think about Lyme patients with their poor immune system and all the other things going on with them. It's not surprising that they're going to pick up germs that are not normally found. But you have to test them, or you're not going to find it. All right, we're going to finish with what I call the testing guide. The IFA, the ILADS, or the Western blot, these serologic tests are not good. They're not recommended because they only look for one species. You get false negatives, false positives. The immunoblot, even though it's a serology test, is much, much better because it uses this lab-created recombinant antibodies and antigens, and it's good for all stages of infection, even early as well as late, very highly accurate. The T-cell assay is, like I said, good only early in the disease and very late, but it can be positive when someone is seronegative with a serology, so it is useful. And I said also it helps to have you figure out the strength of the immune system. The PCR, it detects DNA, fluids, tissues, but it's really very, very insensitive. Like I said, 6 out of 100, so it's not often done by itself. But if you culture first, that's IGeneX's culture, and then do the PCR, then it's at least 10 times, if not more sensitive than that. And the FISH, like I said, is a blood smear. where they actually look for the genetic material of the organism. So it's very, very specific, and it's also good for all different stages. So let's talk about treating. Now, you've heard lectures before, I'm sure. They say, well, you're going to take amoxicillin and you're going to take doxycycline and maybe some tefenoquine, and they talk about all the new things and double dose dapsones and all these other things you've heard about. Well, you know what? That's only a small part of getting better, okay? So let me give an example. You have to, what? Remove toxins. You have to support the mitochondria. You have to support the gut. You have to start then, after you've done this, is when you start treating the tick-borne diseases with antibiotics and herbals. You don't start antibiotics first, okay? You have to also treat the reactivated viruses, the overgrowth of yeasts. You have to get rid of the biofilms. You have to make sure you get enforced rest, because it's when you sleep that your immune system gets recharged, and you definitely need that with a broken Lyme immune system. And finally, and I've lectured on this for decades now, there's a specific exercise program you can do that helps your T cells in your immune system recover. So it's not a program for strengthening or flexibility. It's actually to make your immune system heal, okay? So if you don't do this, none of the antibiotics in the world is going to make you better, okay? So you have to start the basics. All right, detoxification, we'll go through this. I'm going to give you just an example. Different practitioners have their own favorites and their own favorite companies and brands and so forth, but so we'll give you kind of an outline. So what do we do about toxins? It's called mobilize. You mobilize them out of the hiding places, out of the fats, and then once they're mobilized, you bind them and get them out of the body. Okay, so what do? First of all, as I mentioned, glutathione, the detox chemical in our body, gets depleted. So you can take glutathione orally. It's called liposomal glutathione, or you can do it IV. The oral one that I like is Tri-Fortify from Research Nutritionals, but there are others on the market that are good. The next thing is methylation support, even with normal genetics. That helps your metabolism and helps you make more glutathione. And how do you do that? That's methylfolate, hydroxycobalamin, methylcobalamin. Those are the supplements that help to fix that. Liver drainage. Remember I said the bile doesn't drain as well? Well, there are things called ox bile. Believe it or not, it's actual ox bile that you take in a capsule form. There are herbals. One is called Tox-Ease GL by Beyond Balance, Pure Body Clear by IGeneX, and others. These are just examples that help to get the liver activated to make the toxins bind better and get out of the body through that. Now, let's say these toxins are now out of the bile and into your intestinal tract. Well, you don't want them to get reabsorbed and make this little loop around. You want to bind them. So there are binders. There's one called Tox-Ease Bind from Beyond Balance, Mediclay from PRL Lab, Toxin-Pull and Melapol from Research Nutritionals. And what happens is, as the toxins enter your intestinal tract, these binders, which are sometimes made of clay or charcoal and other things, the toxins bind to that and go down the drain with your bowel movement, so they don't get reabsorbed all the time. But as I said, these toxins, once they're in you, will never go away on their own. So detox is one of the things you have to start doing, and it takes a long time. It might take 6 months, may take a year, maybe even 2 years to get all of it out of your system. So please be patient, okay? How do you support the mitochondria? Well, my preferred regimen consists of 3 different products from Research Nutritionals. And again, this is just an example. Other companies have good products too. They have a really high-quality multivitamin called Physicians Daily, oddly enough. A thing called ATP 360 and Energy Multiplex. I recommend all 3 taken together, and you follow the label directions. Often the second, the ATP 360, and the third one, the Multiplex. Usually that's taken until you start to really feel you're recovered. Then you can back off on the dose and maybe even stop them. But the daily one you should stay on probably forever. Now, the mitochondria, as I said, are made of membranes. So you want to do things to help the membranes. Once you take the toxins out, you want to build the membranes back. So that's a compound called phosphatidylcholine, or PC. I get mine from Body Bio, but there are other companies that make it. The mixed omegas, like the omega-3, omega-6, omega-9, those are important too, because they make up the membranes, and that has to be replaced. Extra CoQ10. CoQ10, as you know, is really important for your whole body, and that also lives in the fatty membranes inside the mitochondria. So when we're building back the membranes, you have to add that too. Like I said, other practitioners have their own favorites. This is just an example. I want to tell you about vitamin B6. This is something I learned the hard way. B6 is absolutely vital for normal nerve function. And people often get neuropathy and they get tingles, they get sharp pains and all that. And you take B6. Well, it turns out that's actually bad. B6 is not an active vitamin. It's called a provitamin. It gets converted by your system into the active form called P5P, pyridoxal 5-phosphate. Now, there's a negative feedback loop. If you take B6, the body says, oh, as long as B6 going on, we're not going to convert it into P5P anymore because you don't need it. But that's a paradox because the more B6 you take, the less P5P you have. Okay, so it actually makes you worse. And by taking B6, you can actually become B6 deficient. So basically, if you have neuropathy, which pretty much all Lyme patients do, you should stop every supplement that has B6 in it and switch only to those that have P5P. It's very important. Gut support. Now, the exact regimen I'm not going to go through because it depends on the individual circumstance after testing and so forth. There can be small intestinal bacterial overgrowth. That's what that SIBO stands for. The small intestine should not have a lot of germs in it, but they can overgrow and cause trouble. Could be parasites that are common. Leaky gut. The lining of the intestine gets permeated because it's inflamed and the barrier kind of fails. That can lead to food allergies. Alpha-gal is the allergy to a sugar found in red meats that comes from the tick bites and works through the gut. Achlorhydria, your stomach doesn't make acid anymore and can't digest, can't absorb vitamins. Enzyme deficiencies, all these different things can happen. So whatever regimen you do have to go on, and that's, again, very individual, always you need to have the probiotic support. Okay, used to be take your everything to be fine. Well, that we found out isn't enough. You have what's called prebiotics and probiotics. Prebiotics are things like fiber agents that help the beneficial germs in your body make their good stuff. Okay, and the probiotics are the good germs themselves. So if you're going to take supplements, probiotic supplements, there are a couple of things to look for. One is spore-based organisms. What that means is that in the capsule is not the living germ, but a spore that's like in the dormant state and gets activated in your system. That makes sense because if you're going to get a probiotic in a pill form that's not from a spore, you have to rely on there being living germs in the capsule, which I can't believe is happening. It's been in the capsule for God knows how long. I mean, that's why I have yogurt, because that's a living organism, hopefully. So you want something that's spore-based. You also want to have a compound that has soil-based organisms, because the basis of the good germs in our intestinal tract are the same kind of germs that normally live in healthy soil, interestingly enough. We came from the earth and we are part of it. So you want to have spore-based and soil-based organisms. You want to have a broad spread of beneficial organisms. So what that means is you may have to combine different brains together to get all of this together. And that's fine, but just don't take too much, because if you have a weakened immune system, if you overdose on probiotics, like taking 3 or 4 or 5 a day, you actually can get infected by them. So please don't take too much. Follow the label directions, but just rotate through different compounds, different brands, different types. Sleep is important because it's when you sleep that your immune system gets recharged. So whatever you can do to sleep better, you have to do it. Get rid of the distractions, get rid of all the lights. If you're sensitive to electromagnetic fields, get rid of them if you can. Get comfortable. You get the hot, cold, at night where you're taking off the covers and you cover yourself. But find some happy medium. The sore body, take a pain reliever at bedtime. Usually acetaminophen, a non-steroidal like ibuprofen. If you have a distracting, snoring partner, get rid of them. Make them turn over or go somewhere else. You got to get good sleep. And don't, ignore the fact that something like acetaminophen, and especially the non-steroidals like ibuprofen and naproxen, taken at bedtime can really make you sleep a lot better because you're not having the pain and inflammation so badly during the night. I recommend you avoid narcotics because they actually dampen the sleep response. Even though you're sleeping, you're not getting much benefit out of it. Another great thing, brainwave therapy. We go through different phases of sleep, like stage 1, 2, 3, 4, and then rapid eye movement, and it's all based on the brainwave patterns. And these brainwave patterns you can actually influence by sound. And you can get a free app on your cell phones. I have one that's called Binaural Beats. Write that down. And you put earphones in or headphones like I'm wearing now, and you turn on the thing, and you can set a frequency for relaxation or for sleep or for being alert. And there are very many studies done that show that this really does work. And in fact, on the specific app, there's a help section, and it goes through the theory behind it and the different frequencies you can use and how to use it basically. And I recommend you get that because it really, really is helpful. It's drug-free. It really works. Melatonin. A lot of us get deficient melatonin as we get older, especially as we get sicker. And a little bit of melatonin at nighttime might be helpful. It's also an antioxidant for the brain. The company called Research Nutritionals makes a product called Anxia Ease, anti-anxiety. And believe it or not, eases anxiety, helps you sleep better. And you know what it's made of? It's made of probiotics, because a soothed gut allows you to sleep better. Now, in worst cases, you might actually need a sleeping pill from time to time, and then you get your practitioner to give you what would be appropriate for you at the stage of your infection and so forth. But getting good sleep is important. You have to go through everything you can to sleep a lot. It also means if you're tired in the afternoon, don't fight it. Take a nap. Your body's telling you something. After all this, then you can start the antimicrobials, okay? Now, we recognize nowadays it's not just a simple germ. These germs are complicated. They call them stealth organisms because they hide. They're persistent organisms that persist despite antibiotics. Co-infection is almost universal in a chronic patient. You don't ever find Lyme alone or Babesia alone. There's always some mixture. So the result is most regimens that are successful rely on a combination of antibiotics. Most successful regimens include the botanicals, whether they be a Byron White, Beyond Balance, Nutromedics, whatever your practitioner likes to use. Some people got well with botanicals alone without antibiotics. That can happen, but you have to be very diligent with a really highly trained practitioner and go through a lot of work to get there, but it can work. And like I said, everyone is different. Treatment has to be individualized, and a lot depends on the skill and experience of the practitioner. And the final step in this is to treat the candida and the biofilms in your gut. Now, this is literally the last step because, as I mentioned before, these biofilms are harboring these viruses. So if you right away bust these biofilms in the beginning of treatment, all these viruses are going to come out and make you really a lot sicker. So you have to go through the original steps first to make your body stronger, to get rid of the toxins, to make your immune system better, to make your metabolism stronger, make your immune system, everything better. And then you can start to work on the gut biofilms, okay? You can use different enzymes like Boluc, botanicals like BFM1 from Beyond Balance, but you have to be prepared to treat viruses and yeast because you might get worse from this, from busting the biofilms. So have some antivirals ready, anti-yeast medications, or botanicals ready because that might happen. So that's why I say the best strategy is to hold off on biofilm busting until the end when you have good metabolic support, a lot of detox, and then you go gently. Now, because gut candida is a bad thing, you also have to work on your mouth. Now, why is that? If you have thrush, which is yeast in your mouth, can be candida or any other yeast, every time you swallow, you're going to have it go back in your intestinal tract. So all this biofilm busting and all the anti-yeast stuff and everything is not going to work if you don't clean out your mouth, okay? So how do you do it? Well, it's not normal to have no germs in your mouth, okay? So if you use good antiseptic mouthwash, and I'll show you how to do this in a second, that's going to be great to kill off the yeast that you don't want to swallow it every time you have food. But on the other hand, it's not normal to have no germs in the mouth. So what you do is, as soon as you've done cleaning your mouth with antiseptic mouthwash, to prevent the opportunist germs like yeast from regrowing, that's when you have your probiotics. Research Nutritionals makes one specifically for this, a little tablet that dissolves in your mouth and it releases the probiotics to get your mouth healthy again. Also, if you want to use kefir or yogurt or one of the other ones, it's not just eat it, but swish it around in your mouth a while and swallow it little by little, to get the germs in your mouth first so that the bad germs don't come back. I think the Oramax might be more beneficial than the kefir or the yogurt, but use them both. Now, what antiseptic? And this is really going to surprise you. Believe it or not, it's a thing called Dakin solution, which is, believe it or not, Clorox, very highly diluted household bleach. Now, no, I'm not losing my mind, but this is actually something you can do. It's got to be super diluted. We're talking about a teaspoon in 4 ounces of water. So it's kind of like a commercial swimming pool. It's not really very strong. But the idea is that you put some of it in your mouth and you hold it in your mouth while you're brushing with a toothbrush, your tongue, your gums, your cheeks, your palate, your teeth, everything. You scrub it really, really well. Then you spit it out, put water in your mouth, and do the scrubbing again to now rinse it. And that's when you have the Oramax or the Kefir or both, okay, to put the good germs back in. Now, this is not meant to be on a regular basis. It's usually a one-time treatment. You might have to repeat it again X number of weeks or months down the line, but it's definitely not something you can do on a very regular basis because there's no information on what this will do to your teeth over time. I mean, it might dissolve some of the minerals out of your teeth, so it's not meant to be a regular thing. This is like a drastic thing to just get ahead of it, one-time shot, and then you're going to be so much better. Right away, you feel your intestinal tract calms down. And finally, like I said before, is the exercise regimen. And I'll tell you this again, no matter what you do with your treatment, you will not completely recover if you don't exercise. It's a very specific regimen, and it's there to support your immune system. It's not intended to make you stronger or make you have better stamina, although that probably will happen too. And what this is, it's a whole body toning and stretching program. It's absolutely not aerobics, because aerobics, interestingly, can make you worse, because aerobics have a negative effect on the immune system. And in fact, another fact of this is that the exercise cannot be every single day, because what exercise does, it dampens the immune system for a day or so, and then the day after, it rebounds stronger. So the protocol calls for an exercise not every day in a row, but like once, and then you'll have 1, 2, or 3 days of rest. And then you do the exercise, then several days of rest. And the exercise, like I said, is not aerobic, because aerobic worsens this dip in your immune system. Here are the specifics. Number one, it's a whole body regimen. Now, some muscle guys, they'll go to the gym and they'll work on their arms one day, and then they'll work on their legs the next. No. To get over Lyme, what you have to do is the whole body, where you might start from the head and work yourself down to the toes. So you're going to do a little exercise of every part of your body over the session that you're there. And it consists of using very light weights, like maybe a 1 or 2 pound weight, or maybe some resistant bands or some machines with very light weights on it. So the idea is very light weights, but a lot of repetitions. And you begin very gently, and let's say you worked on your arms, then before you go to the next part of your body, you stretch that arm. Then you go, let's say, to your legs. Then after you're done, you stretch the legs before you move to the next, because stretching is really important to get your body mobilized, get the toxins out, to stretch your joints. If you look up exercise programs, this is called stretch and tone and body sculpting. It's a modification of that. Now, this is another trick. After the exercise, immediately have a hot bath or hot shower or sit in the sauna, because you want the heat to continue the process of rebuilding the immune system. As soon as you're done with that, then you really should lie down and rest. And you're going to find that when you first do this, you're really going to want to lie down and rest. But, you know, over a period of time, maybe 6 weeks or so, you're going to come out of the exercise saying, I'm feeling energized. I'm not going to lie down now. I'm finally feeling good. And that means you're starting to help to heal your immune system. Now, over time, you know, you can make the 1 pound weight become a 2 pound weight, you know, that kind of thing. But you're never going to go and do aerobics, and you're never going to exercise every day. Okay, so it's going to be a day of exercise and maybe 2 days off. And as you get better, maybe a day of exercise and 1 day off. But never daily, and never aerobics until you're completely well and you can do what you want. And so that's it. I didn't run over time too much. So it's simple, right? So self-awareness, in other words, pay attention to your body, keep a diary, see what's really going on. Educate yourself, do these seminars, get your support group to do this more often if you can. Keep your diary, keep all copies of your medical record. You know, so many times your care gets fragmented and your records get lost. So anything that's done, even if it's, you know, electronic, download it, print it out, put it in a file, keep your own. Try not to doctor shop. Find a good practitioner, stick with that person, because over time they'll get to know you better, and the treatment will evolve, and hopefully you'll have a good outcome. And like I said before, insist upon regular visits, always schedule follow-up, and attitude. Always be optimistic and positive, because you know you can get better. And one thing is, a lot of patients with Lyme, they'll have good days and bad days, and say, well, I felt really good the other day, now I'm not feeling so well. Well, hold on to that. The good day means you can get well. It could be good all the time. It means it's not permanent, okay? So optimism here is really called for, because it's true. You can get better. And now I am done. So I'm going to stop the screen share. We can talk. All right. Is everyone still here and awake?
Just a note, I muted everybody, so if you want to speak, you're gonna have to unmute yourself.
And feel free to ask a question, or if you want to type it in the chat, that's fine too.
Is it okay if I raise my hand?
Sure.
Yeah. Well, here's that question. When Dr. Burrascano was talking about the tests, I was interested in the one that you can do at home to get a kit and do a finger stick. Can you use that to get a culture test?
Well,
'Cause the
it's
culture test-
basically a pared down version of the immunoblot. You know, an immunoblot gives a lot of details. It tells you IgM, IgG, the different species and so forth. This is a very pared down immunoblot. It just says positive or negative, yes or no. So it's a very simple at-home test. It's not a culture. The culture requires kind of a large volume of blood. And I'll give you a hint. You know, the kit for getting culture is a tube. Send more than one tube, because the more blood they have, the more chance they are picking up the infection.
Yeah. Well, I've never had a culture before, but I would like to know a larger sample, what's in there.
Yep,
Thank
culture is
you.
a good idea. Yep. Well, let me ask you guys a question. How is everybody? Are you all Lyme patients? Have you been doing well, not so well? Tell me your stories.
I have a question.
Okay.
I'm a complicated patient, and I was bit, just to tell you a little bit. I was bit in 2012, had the bite, never diagnosed, never treated, started having symptoms, autoimmune, was always able to function though. Later, I found out years later, worked in a moldy building. I got really sick. Eventually, just recently had some testing done. I'm positive for the HDLAR gene, which is the mold susceptibility. And so I have mold illness, and I was living in mold, and I've worked in mold. And then I got Epstein-Barr virus in 2019. And they gave me antibiotics, not knowing I had Epstein-Barr, and it just kind of triggered everything. And then I had a pulled tooth in 2021. It got infected. I still didn't know I had Lyme. I suspected it, but never treated. And they put me on antibiotics for 4 months. I got C. difficile infection and then 3 COVIDs in 1 year in 2022.
Wow.
I have not
Jackpot.
I have not been the same, and I am positive with vibrant testing. Borrelia myomytoi, Afzeli, Garninia. I don't know if I'm pronouncing those correctly. My Epstein-Barr virus is activated. My early antigen is positive. Everything's just activated. I mean, I've literally been homebound sick and for 2 years now, and trying to do all these steps. And I think I'm finally getting into the right doctor, but I have a lot of detox SNPs where I can't detox. I just did a GI map test. I have some fungus, and I also have Citrobacter, and 2 other bacterias in my gut that are showing. And so this doctor, I think, is going to try to start with some of that stuff and try to stabilize me before we start on the Lyme. I also have Babesia, Duncani, and toxoplasmosis. So I'm really sick, and I'm just learning more all about this stuff and the order to do everything. And I don't know if someone like me, where I got C. difficile and I know my gut isn't completely healed, if I would even be there in time to do antibiotics, or if someone like me is better to do herbals, and what treatment do I start on first, where I'm learning from you, like the yeast and the biofilms, you probably wait to do that at the end.
So, you know, the lecture I gave today, I think, was very key to the kind of things that you've experienced in so many other Lyme patients, because you go down this cascade, you know, you get a little bit sick, or don't even recognize you're sick from a tick bite, and then little by little things get worse, and then this whole thing blows up. then COVID comes along and throws a real monkey wrench into it. And I just want to tell you, a study's being done in New York City on patients in the long COVID clinic, and they found tick-borne disease in 70% of them. So long
Wow.
COVID is probably reactivation of Epstein-Barr and all that, but also tick-borne disease. So I think the lecture I gave you tonight, I can send the slides to Johan and he'll give you a copy if you want. And you can actually use that as your roadmap, because this is a step-by-step process. And, you're going to need to do detox. You're going to need to support your metabolism and all the basic things I mentioned. And so I'm glad I gave you this lecture tonight rather than just talk about tests.
So and what was that? You will give me a what? A copy of what to?
The slides I gave tonight, that I presented tonight, you can get a copy of them from Johan. He'll send it to you by email.
Yeah,
Oh, perfect. Okay.
I'll send it out through Britt, so I'll send the PowerPoints to her and then she can share it with the group.
Okay.
Actually, I updated the PowerPoint tonight, so I'll give you a new one. Don't send the one you
Perfect.
have.
Are you okay if we post those, this recording and the PowerPoint slides on our website for the Kansas City area?
I have no objection.
Okay, thank you.
Dr. Burrascano, I have a question.
Sure.
Could I just jump in quickly? Would everybody feel more comfortable if I stopped recording before we ask these questions? Is that appropriate?
That's fine.
Fine with me.
Okay, why don't I do that? Because you may not want this all,